Margaret Chen had lived in her small Oregon farmhouse for nearly 40 years. At 73, she still enjoyed working in her garden, though her knees complained more than they used to. The property sat at the edge of a dense forest, where tall pines cast long shadows across her vegetable patches and flower beds. She loved the quiet out here, far from the noise of the city she'd left behind when she retired from teaching botany at the local college. That particular Saturday morning started like any other. Margaret had her coffee on the porch, watching the mist roll through the trees. She'd been meaning to clean out her garden shed for months now. The small wooden structure had become a dumping ground for old pots, rusty tools, half-empty bags of soil, and all sorts of things she'd convinced herself she might need someday. Her daughter had been nagging her about it during their weekly phone calls, worried that her mother might trip over something in the cluttered space. So after breakfast, Margaret pulled on her gardening gloves and headed to the shed with a determination she didn't quite feel. The door creaked as she pushed it open, and dust particles danced in the shaft of morning light that cut through the dimness. She sneezed twice and made a mental note to wear a mask next time. The shed smelled of earth and wood and something else she couldn't quite place, something almost sweet. She started with the shelves along the left wall, pulling down clay pots and checking them for cracks. Most were fine, just dirty. Margaret stacked them in a box to wash later. Then she moved to the back corner where she'd piled up bags of fertilizer and old seed packets. As she bent down to grab a particularly heavy bag, something made her pause. It was a sound, or maybe the absence of sound. The birds outside had gone quiet. Margaret straightened up slowly, her back protesting. That's when she noticed it, high up in the corner near the rafters, hanging from an old hook she'd forgotten was even there, was something that absolutely should not have been in her shed. At first glance it looked like a giant wasp nest, but as her eyes adjusted to the shadows, she realized it was something entirely different. The thing was huge, easily the size of a basketball, maybe bigger. It hung there like some kind of alien egg, suspended by what looked like thick silk threads. But what really caught her attention was the surface. Even in the low light, she could see it had a strange sheen to it, almost like oil on water. Colors seemed to shift across its surface, purples and greens and golds that shouldn't have been there. Margaret had spent her entire career studying plants and insects. She'd seen cocoons before, plenty of them. Monarch butterflies, luna moths, all sorts of local species. But this was completely wrong. Everything about it screamed impossible. No insect in Oregon grew to that size. No cocoon she'd ever seen had that kind of surface texture or those colors. She took a step closer, her scientific curiosity overriding the slight unease creeping up her spine. The structure had an almost leathery appearance, tough and thick. But there were patterns on it too, intricate designs that looked almost deliberate. They reminded her of fractals, those endlessly repeating mathematical patterns found in nature. Except these seemed too perfect, too organized. Margaret noticed something else. The air around the cocoon felt different, warmer somehow. She held her hand up near it, careful not to touch, and yes, definite warmth. That wasn't normal either. Most cocoons were dormant structures, their inhabitants in a state of suspended animation. They shouldn't be generating heat. She stood there for a long moment just staring. Part of her wanted to poke it with a stick to see what would happen. But the more sensible part, the part that had spent decades in laboratories and field research, told her to leave it alone and call someone who could help figure out what this thing was. Margaret backed out of the shed carefully, not taking her eyes off the cocoon until she had to. She pulled the door closed and headed straight for the house. Her hands shook slightly as she flipped through her old address book, the one she kept by the kitchen phone. Most of her former colleagues had retired too, scattered across the country. But she remembered that James Whitmore had taken over the entomology department at Oregon State. They'd worked together years ago on a project studying native moth populations. She dialed the number and waited through 4 rings before a young voice answered. The department secretary, probably a student worker. Margaret explained who she was and asked to speak with Dr. Whitmore. There was a pause, some typing, and then the secretary said he was in a meeting but could call back. Margaret said it was urgent, that she'd found something unusual and needed his expertise. 10 minutes later, her phone rang. James sounded older than she remembered, his voice a bit rougher, but he still had that same enthusiasm when she described what was in her shed. At first, he seemed skeptical. Oregon didn't have insects large enough to create cocoons that size. But when she described the colors and the warmth, his tone changed. He asked if she'd touched it. She said no. Good, he told her. Don't touch it, don't disturb it, and definitely don't try to open it. He'd drive out that afternoon with a couple of his graduate students. Margaret spent the rest of the morning trying to distract herself with housework, but she kept glancing out the window toward the shed. She made lunch, but barely ate. What was that thing? Where had it come from? Her shed wasn't sealed perfectly, but it seemed unlikely that something could have gotten in, created that massive structure, and she'd never noticed. How long had it been there? She thought about the sweet smell she'd noticed when she first opened the shed. Had that been coming from the cocoon? And the patterns on its surface, those fractal-like designs. They reminded her of something she'd seen in a textbook once, but she couldn't quite remember what. When James finally arrived, he had 3 people with him instead of the 2 he'd mentioned. They came in a university van loaded with equipment. James looked much the same as Margaret remembered, just grayer and a bit rounder. He introduced his team quickly, barely able to contain his excitement as Margaret led them to the shed. She opened the door and stepped aside, letting the scientists enter first. Their reactions were immediate and gratifying. James let out a low whistle. One of the students, a young woman with bright red hair, actually gasped. They stood there in a cluster, all 4 of them just staring up at the cocoon. James was the first to speak. "Margaret," he said slowly, "in 30 years of studying insects, I've never seen anything like this." He pulled out a camera and started taking photos from every angle. Another student set up some kind of sensor on a tripod, aiming it at the cocoon. The 3rd member of the team was already unpacking what looked like collection equipment. Margaret watched from the doorway as they worked. They moved carefully, methodically, documenting everything. James held up a thermometer near the cocoon and shook his head in disbelief. 3 degrees warmer than ambient temperature, he told the others. One of the students suggested it might be some kind of invasive species, something brought in from another country. But James didn't think so. The construction was too unusual, the materials too unique. They worked for nearly 2 hours, taking measurements and samples of the silk threads attached to the rafters. Nobody suggested cutting the cocoon down, not yet. James wanted to do this properly, with proper protocols and safety measures. Whatever was inside that thing, it deserved to be studied carefully. As the sun began to set, James pulled Margaret aside. "We need to take this back to the lab," he said. "I want to run tests, maybe do some imaging to see what's inside without opening it. Would you be comfortable with that?" Margaret nodded, of course. That thing didn't belong in her shed anyway. She wanted answers just as much as they did. They scheduled a return visit for the next morning with more equipment and a proper container for transport. As the van pulled away down her long driveway, Margaret stood in her yard looking back at the shed. The door was closed now, the cocoon hidden again in the darkness. But she couldn't shake the feeling that something profound was waiting inside that strange, shimmering structure, something that would change everything. The next morning arrived with a light drizzle, typical for Oregon in late spring. Margaret had barely slept, her mind racing with possibilities about what might be inside that cocoon. She'd spent half the night reading old textbooks and searching through her files from decades of field research. Nothing matched. Nothing even came close. James and his team showed up at 8 o'clock sharp, this time with even more people and equipment. The university van was joined by a smaller truck carrying what looked like a climate-controlled transport container. Margaret counted 7 people total, including James. They'd brought in specialists, he explained. People who knew about insect biology, material science, even a veterinarian who specialized in exotic species. They gathered in Margaret's kitchen first, spreading out papers and tablet computers across her dining table. James wanted to brief everyone on what they'd found yesterday and outline the plan for today. Margaret made coffee and listened as they discussed extraction procedures and safety protocols. One of the new arrivals, a woman named Dr. Patricia Lopez, kept asking questions about the temperature readings and the structural composition of the cocoon's outer layer. The plan was straightforward but careful. They'd set up full documentation equipment first, video and still cameras from multiple angles. They'd take additional temperature and humidity readings, air samples from around the cocoon, and try to get more detailed measurements of its exact dimensions. Then, if everything checked out and it seemed safe, they'd carefully detach it from the rafters and place it in the transport container. Margaret followed the group out to the shed, holding an umbrella against the drizzle. The scientists moved with practiced efficiency, setting up their equipment in the cramped space. Lights were positioned to eliminate shadows. Cameras on tripods were placed at 3 different angles. Someone set up a laptop on an overturned bucket, its screen showing multiple camera feeds. Dr. Lopez approached the cocoon with what looked like a medical stethoscope. She pressed it gently against the surface and listened intently, her eyes closed in concentration. After a moment, she opened her eyes and looked at James with an expression Margaret couldn't quite read. "It's vibrating," Dr. Lopez said quietly. Very subtle, but definitely there. Some kind of internal movement or process. That changed the atmosphere in the shed immediately. If something was moving inside, it might be close to emerging, or it might be dying. Either way, they needed to move faster. James made a quick decision. They'd take the cocoon now, get it back to the lab where they had better equipment and controlled conditions. 2 of the graduate students climbed up on a ladder they'd brought, working carefully to detach the silk threads from the old hook and rafters. The cocoon swayed slightly as they worked, and Margaret found herself holding her breath. The material proved tougher than expected. They needed wire cutters for some of the thicker anchor threads. As they lowered the cocoon into a padded carrier, Margaret noticed something she'd missed before. The bottom of the structure had tiny holes almost like breathing pores. They were arranged in a perfect circular pattern, and when she mentioned it to Dr. Lopez, the scientist immediately took close-up photographs. "Good eye," Dr. Lopez said. "Those might be ventilation structures. Whatever made this needed air exchange." The transport container looked like something you'd use to move an organ for transplant. It had clear sides, temperature controls, and humidity monitors. They settled the cocoon inside, padding it with foam and securing it so it wouldn't shift during the drive. Someone placed wireless sensors inside the container, tiny devices that would monitor any changes in temperature, movement, or gas composition. Before they left, James asked Margaret if she'd be willing to come to the lab later in the week. They might have questions about the shed, about when she last cleaned it, about anything unusual she might have noticed in recent months. "Of course," Margaret said. She wanted to know what they found out anyway. James promised to call her with updates. After they left, Margaret stood in her empty shed for a long time. The space where the cocoon had hung looked oddly bare now, just an old hook on a support beam. But the warmth was gone too, she realized. The shed felt normal again, just a simple garden storage building. Whatever that thing had been, it had definitely been generating its own heat. At the university laboratory, the team worked through the afternoon setting up a proper observation station. The cocoon was placed in a specialized chamber, one normally used for studying sensitive biological samples. The room could be darkened completely or lit with different wavelengths of light. Temperature and humidity could be controlled precisely. And most importantly, cameras would record everything 24 hours a day. James called Margaret that evening with the first round of results. The spectroscopy readings were bizarre, he told her. The cocoon's outer layer contained chemical compounds they couldn't immediately identify. There were trace elements present that shouldn't be there, things that weren't part of any known insect's biology. The silk threads were exceptionally strong, stronger than spider silk, which was already one of the toughest natural materials known. But the really interesting part was what the imaging revealed. They'd used a portable ultrasound machine, the kind doctors use to check on babies before they're born, to try to see inside without damaging the cocoon. The images were unclear, but there was definitely something in there. Multiple somethings, actually. The structure wasn't hollow with a single large organism inside. It was more like a complex arrangement of smaller components all interconnected. Over the next few days, Margaret received regular updates. The cocoon remained stable in its controlled environment. The subtle vibrations Dr. Lopez had detected continued, following a rhythm that seemed almost tidal, stronger at certain times of day and weaker at others. The temperature it generated remained consistent, always about 3 degrees above ambient. On the 3rd day, something changed. One of the graduate students monitoring the overnight camera feeds noticed it first and called James at 6 in the morning. The colors on the cocoon's surface were shifting more rapidly now. The patterns were moving, flowing across the leathery exterior like slow-motion waves, and there was a new sound, barely audible even with sensitive microphones, a kind of soft clicking or tapping from inside. By the time Margaret arrived at the lab later that morning, the observation room was crowded with people. Word had spread through the university's science departments, and researchers from biology, chemistry, and even physics had found excuses to stop by. James had to limit access to essential personnel only. Too many people would compromise the environmental controls. Margaret stood behind the observation window, watching the cocoon through the glass. Even from this distance, she could see the surface shimmer and shift. It was beautiful in a strange, unsettling way, like looking at something that didn't quite belong in the normal world. James stood beside her, making notes on a tablet. "We're getting inquiries from other universities," he told her. "Everyone wants to know what we found. I've been vague so far, but we can't keep this quiet much longer. If this turns out to be a new species, or worse, some kind of invasive organism, we need to notify the proper authorities." Margaret understood, but she felt oddly protective of the thing. It had been in her shed, hidden and safe, and now it was under bright lights with dozens of strangers studying its every movement. She kept those thoughts to herself, though. This was science, and science required observation and documentation. That afternoon, they detected the first crack. It was tiny, barely visible, running along one of the seams where the surface patterns met. Dr. Lopez spotted it during a routine inspection and immediately called everyone over. The crack wasn't growing, not yet, but its appearance suggested something was about to happen. James made another decision. They'd watch it around the clock now. Teams of 2 would take shifts, always someone in the observation room. If the cocoon started to open, they needed to be ready to document everything. He asked Margaret if she wanted to stay, maybe take a shift. She agreed immediately. She'd come this far. She needed to see what happened next. The 4th day passed with agonizing slowness. The crack didn't grow. The vibrations continued. The colors kept shifting. Margaret took the evening shift with Dr. Lopez, both women sitting in the dimly lit observation room, watching the cocoon like it was the most important thing in the world. And maybe, Margaret thought, it was. They talked quietly about possibilities. Dr. Lopez had a theory that it might be some kind of communal structure, multiple insects working together to create a shared cocoon for metamorphosis. Margaret had read about similar behavior in some tropical species, but nothing on this scale. James thought it might be a previously unknown life stage of a known species, something that only happened under specific conditions that humans had never observed before. On the 5th day, everything changed. Margaret was home trying to catch up on sleep when her phone rang at 3 in the afternoon. It was James, and his voice was shaking. "You need to get here now," he said. "It's opening." Margaret drove faster than she probably should have, her heart pounding. When she reached the lab, there were people everywhere. Security was trying to keep unauthorized personnel out of the building. She showed her temporary access badge and rushed to the observation room. Through the window, she could see the cocoon had split open along multiple seams. The outer layer was peeling back like flower petals, revealing something inside that glowed with a soft, eerie light. It was blue-green, pulsing gently, and as Margaret watched, she realized she was seeing something that no human had ever witnessed before. Not a new species. Something far more incredible than that. Something that would change everything they thought they knew about life, death, and survival. James stood beside her, speechless, as the cocoon continued to open. The light grew brighter, and in that moment, Margaret understood that her simple Saturday morning shed cleaning had led to one of the most important scientific discoveries of the century. The observation room had gone completely silent. Even the usual hum of equipment seemed muted as everyone stared at the cocoon through the protective glass. Margaret pressed closer to the window, her breath fogging the surface. Behind her, she could hear the soft clicks and whirs of cameras recording every second of what was unfolding. The outer layers of the cocoon continued peeling back in slow motion, revealing more of that strange glowing interior. The blue-green light pulsed in waves, growing stronger and then dimmer, like a heartbeat made visible. Dr. Lopez had her face pressed to a specialized viewing scope, taking notes rapidly on her tablet without looking away from the eyepiece. "The bioluminescence is unlike anything I've seen," she murmured. "It's not following typical patterns. There's structure to it. Organization." James was on his phone, speaking in hushed, urgent tones with someone from the National Science Foundation. Margaret caught fragments of his conversation, words like unprecedented and requiring immediate funding. But she couldn't focus on that. Her eyes were locked on the cocoon. As more of the outer shell fell away, the structure inside became clearer. What they were seeing wasn't a single organism at all. It was a network, an intricate web of what looked like crystalline threads interwoven with organic material. The threads glowed from within, creating that eerie light they'd first noticed. And attached to these threads, suspended throughout the interior like ornaments on some alien Christmas tree, were dozens of small, delicate forms. One of the graduate students gasped. "Are those wings?" Margaret squinted, trying to make sense of the shapes. The student was right. Those delicate forms had wings, or what remained of wings. They were moths, she realized with a jolt. Dead moths, their bodies perfectly preserved within the glowing network. But they weren't just randomly scattered. They were arranged in a specific pattern, almost ceremonial. "We need to collect samples of that luminescent material," James said, putting his phone away. "But we have to be careful. If this is some kind of fungal growth, we need to know if it produces spores, if it's potentially hazardous." Dr. Lopez stepped back from her viewing scope. "I think it is fungal," she said slowly. "But I've never seen anything like this structure. It's using the moth bodies as anchor points, but it's also doing something else. Those threads are connected to the moth's remains in very specific places, almost like it's accessing their tissue." A chill ran down Margaret's spine. She'd studied plenty of parasitic relationships in nature during her career. Fungi that took over ants' brains, making them climb to high places before killing them. Wasps that laid eggs inside caterpillars. Nature was full of horror stories if you looked close enough. But this felt different. This felt almost purposeful, designed. Over the next hour, the cocoon opened fully. What they'd thought was a simple container for metamorphosis was actually something far more complex. The glowing fungal network filled the entire interior. But it wasn't just random growth. It had structure, architecture. The threads formed chambers and passages, all centered around the preserved moth specimens. James suited up in protective gear to enter the containment chamber. He moved slowly, carefully approaching the opened cocoon with a collection kit. The rest of the team watched through the window as he used sterile tools to extract a small sample of the glowing material. Even through the glass, they could see how it clung to the collection swab, stretching like melted cheese before finally breaking free from the main mass. When James emerged from the containment chamber, he held the sample container up to the light. The material inside still glowed, pulsing gently. "Let's get this under a microscope," he said. "And we need to run a full genetic analysis. Whatever this is, we need to identify it." The next few hours were a blur of activity. The fungal sample was divided and sent to different labs within the building. Microscopy revealed an incredibly complex cellular structure, unlike typical fungi. The DNA analysis would take longer, but preliminary results started coming back by evening, and that's when things got truly strange. The fungal DNA contained sequences that matched known species in the database, but it also contained something else entirely: genetic material that the computers flagged as belonging to insects. Specifically moths, more specifically a species called Pseudonarda flava, according to the database. But when Dr. Lopez looked up that species name, her face went pale. "That can't be right," she said, showing Margaret and James her computer screen. "Pseudonarda flava has been extinct since 1987. It was declared gone after its habitat in Northern California was destroyed for development. The last confirmed sighting was almost 40 years ago." Margaret felt her knees go weak. She sat down heavily in a nearby chair. "The moths in the cocoon," she said quietly. "Could they be?" "We need to extract one of the specimens for identification," James said immediately. "If those are actually Pseudonarda flava, if there's any viable genetic material, this changes everything." They worked through the night. James and Dr. Lopez carefully removed one of the moth specimens from the glowing network. It came away reluctantly, the fungal threads clinging to it. Under bright lights, the moth was beautiful despite being clearly long dead. Its wings had a delicate yellow coloring with brown markings. Margaret photographed it from every angle, while Dr. Lopez took tissue samples. The DNA results came back at 3 in the morning. Margaret was dozing in a chair when James shook her awake. His eyes were red from exhaustion, but they gleamed with excitement. "It's confirmed," he said. "Pseudonarda flava. Margaret, you found a colony of extinct moths in your shed. Somehow they survived hidden away. And this fungus, it's been preserving them." But there was more. As dawn broke and more analysis came in, They began to understand the full scope of what they'd discovered. The fungus wasn't just preserving the dead moths, it was incorporating their DNA into its own structure. The genetic sequences they'd found weren't contamination. They were intentional, part of the fungus's own genome. It's like the fungus consumed them, Dr. Lopez explained to the growing crowd of researchers who'd gathered for an early morning briefing. But instead of just breaking down their tissues for nutrients, it archived their genetic information. It's storing the blueprint for an extinct species inside its own DNA. Someone asked the obvious question. Why? What's the evolutionary advantage? Nobody had an answer. Not yet, anyway. But Margaret kept thinking about something she'd noticed during her initial discovery. The sweet smell in the shed. The warmth around the cocoon. Those weren't just random byproducts. The fungus had been actively maintaining an environment. It had been keeping something alive, or at least preserved in the best way it could. Over the following days, they made more discoveries. The fungal species was entirely new to science, never before documented. They decided to name it Luminomyces symbioticus for its glowing properties and its apparent relationship with the moths. But the more they studied it, the more questions arose. How had the moths and fungus come together in the first place? How long had they been in Margaret's shed? And most intriguingly, why had the fungus preserved not just one or two moths, but dozens of them, all arranged in that careful pattern? One of the graduate students had a theory. She'd been studying the arrangement of the moths within the cocoon structure and noticed something. They were organized by age. The oldest specimens were at the center, the youngest near the outer edges. It was like a timeline, a chronicle of a dying population. The last few members of an extinct species had somehow found their way to Margaret's shed, possibly attracted by some chemical signal from the fungus. And instead of letting them disappear completely, the fungus had preserved them. But why? James kept asking. Fungi don't think. They don't make conscious decisions to preserve other species. This has to be some kind of automatic process, something that evolved for a specific purpose. Dr. Lopez had been thinking about that. What if it's about survival? She suggested. The fungus preserves species that are dying out, incorporates their DNA. Maybe in some environments, having that genetic diversity archived within its own structure provides some kind of advantage. Maybe it can access those genes somehow, use them when environmental conditions change. It was a radical idea, something that would challenge fundamental understanding of how organisms interact and evolve. But the evidence was right there in front of them, glowing softly in its containment chamber. A fungus that collected and stored genetic information from other species like a biological library. Margaret stood at the observation window late one night, alone except for the monitoring equipment. The cocoon structure still glowed, though dimmer now that it had been opened and sampled. The moths inside, those last remnants of a lost species, seemed almost peaceful in their crystalline tomb. She thought about all the species that had gone extinct in her lifetime, the ones she'd read about in scientific journals, mourned quietly, and then moved on from because what else could you do? But maybe this discovery meant something. Maybe it meant that extinction wasn't always as final as they'd thought. Behind her, the door opened quietly. James entered carrying 2 cups of coffee. He handed her one and stood beside her at the window. "Can't sleep either?" he asked. "Too much to think about," Margaret replied. "James, what if there are more of these? More fungal colonies preserving extinct species?" "That's exactly what I've been wondering," he said. "I've already put in requests to search the areas where Pseudonarta flava used to live, and I'm reaching out to colleagues around the world, asking if they've ever seen anything similar. If this is happening here, it might be happening elsewhere." They stood in comfortable silence, watching the soft pulse of bioluminescent light. The cocoon that Margaret had found in her dusty shed had opened up more than just questions about one extinct moth species. It had opened up the possibility that life found ways to persist that humans had never imagined. That survival could take forms so strange and wonderful that even dedicated scientists had missed them completely. And somewhere in that glowing network of fungal threads and preserved wings, in the DNA sequences that blurred the line between species, there was a message. Life didn't give up easily. Even in the face of extinction, it found ways to hold on, to remember, to hope for a future that might allow lost things to return. The news broke on a Wednesday morning, exactly 3 weeks after Margaret had first opened her shed. James had tried to keep the discovery quiet while they completed their initial research, but someone leaked it to a science journalist. By noon, every major news outlet was reporting on the extinct moths found preserved in a mysterious glowing cocoon in Oregon. Margaret's phone rang constantly. Reporters wanted interviews. Documentary producers pitched shows. Even a Hollywood agent called, suggesting they could turn her story into a movie. She unplugged her landline and let most calls go to voicemail on her cell phone. The attention was overwhelming. At the university, security had to be increased around the lab building. People showed up wanting to see the cocoon. Everyone from curious locals to professional scientists who'd flown in from other countries. James organized a press conference hoping it would satisfy some of the curiosity and let them get back to work. Margaret reluctantly agreed to attend. The press conference room was packed. Cameras lined the back wall. Reporters filled every seat, their recorders held up like offerings. Margaret sat at a long table with James and Dr. Lopez, feeling completely out of her element. She was just a retired botanist who'd found something strange in her shed. She never asked to become the center of an international science story. James handled most of the questions, explaining the discovery in terms that non-scientists could understand. Yes, the moths were definitely Pseudonarta flava, a species that had been declared extinct nearly 4 decades ago. Yes, the fungus was previously unknown to science. No, they didn't yet understand exactly how or why the fungus preserved the moth's genetic material. That would take more research. Someone asked the question Margaret had been dreading. What does this mean for conservation efforts? If extinct species can be preserved this way, does it change how we think about extinction? James paused, choosing his words carefully. It's too early to say definitively, but this discovery does suggest that our understanding of extinction may need updating. Just because we can't find living specimens of a species doesn't necessarily mean all trace of them is gone. There may be natural preservation mechanisms we haven't discovered yet. After the press conference, Margaret escaped back to her farmhouse. But even there, things had changed. News vans parked at the end of her long driveway. A few persistent reporters tried to walk up to her house before the local sheriff convinced them to leave. Margaret felt like a prisoner in her own home. Meanwhile, at the university lab, research continued at an intense pace. The initial findings about Luminomyces symbioticus raised more questions with each passing day. Dr. Lopez led a team analyzing the fungus's genetic structure, trying to understand how it had evolved this remarkable preservation ability. What they found was extraordinary. The fungus had genetic sequences from at least 7 different insect species in its DNA, not just the moths. Some of those sequences matched known extinct or endangered species. Others didn't match anything in existing databases, suggesting they came from species that had died out before humans ever documented them. The fungus was like a living archive, a biological museum preserving fragments of lost biodiversity. But how did it work? That was the question keeping researchers up at night. Dr. Lopez and her team discovered that the fungus had specialized structures in its cells, almost like tiny storage compartments where it kept the foreign DNA sequences organized and intact. It was protecting that genetic information from decay, maintaining it with the same care a librarian might preserve ancient manuscripts. Even more fascinating, they found evidence that the fungus could actually express some of the stored genetic information. Under certain conditions, it produced proteins coded by the moth DNA, proteins that moths would normally use for things like wing development or pheromone production. The fungus was borrowing genetic tools from the species it had archived. It's like horizontal gene transfer on steroids, Dr. Lopez explained during a team meeting that Margaret attended via video call. Bacteria swap genes all the time, but this is a fungus taking large, functional genetic sequences from complex organisms and integrating them into its own biology. If we can understand the mechanism, it could revolutionize genetic research. The implications went far beyond academic interest. If they could understand how the fungus preserved DNA so effectively, it might lead to better ways to store genetic samples in seed banks and conservation programs. If they could figure out how it selected which genetic information to preserve, it might reveal something about what traits were most important for survival in changing environments. James received a major grant from the National Science Foundation to expand the research. The university built a new dedicated lab space just for studying the cocoon and the fungal samples. More researchers joined the project, bringing expertise in genetics, evolutionary biology, ecology, and even computer science to help model the fungus's behavior. Margaret visited the lab regularly, Though she stayed away from the media attention as much as possible, she'd become something of a scientific celebrity despite her discomfort with fame. Other researchers treated her with a mix of respect and curiosity. She was the person who'd made the discovery, even if it was largely by accident. One afternoon, about 2 months after the initial finding, James called her with news. They'd found another cocoon structure. Not in Oregon, but in Northern California, in one of the last remaining patches of habitat where Pseudonarta flava used to live. A team of researchers searching the area had discovered a smaller cocoon in an abandoned barn, this one only about the size of a softball. But it had the same glowing interior, the same preserved moth specimens. "We're organizing an expedition to search more systematically," James told her, "not just for this species, but for any similar fungal structures. If this is happening in multiple locations, if the fungus is more widespread than we thought, we need to map it, study it, understand how common this preservation mechanism really is." Margaret wanted to go on the search, but her daughter convinced her to stay home. The terrain would be rough, the work physically demanding. At 73, Margaret had to admit she wasn't up for hiking through forests and poking around in abandoned buildings. But she followed the expedition's progress closely through daily video calls with James. They found 5 more cocoon structures in that first month of searching. Each one was different. One contained the preserved remains of a butterfly species thought to be extinct. Another had beetles. A third, largest cocoon held what appeared to be the last members of a native bee population that had vanished from California decades ago, probably wiped out by disease or pesticide exposure. Each discovery reinforced the same pattern. The fungus Luminomyces symbioticus seemed to appear in the final habitats of dying species. It preserved the last survivors, incorporating their genetic information into its own structure. It was acting like a biological emergency backup system, automatically archiving genetic diversity that would otherwise be lost forever. The scientific community was electrified. Papers were published in rapid succession. Researchers began finding similar fungal species in other parts of the world, though none exactly like the one Margaret had discovered. A team in Madagascar found a fungus preserving endemic frog species. Researchers in Australia discovered one working with extinct marsupials. Each variant had slightly different properties, but the basic mechanism was the same. Dr. Lopez made another breakthrough discovery about 4 months into the research. She'd been studying the fungus's reproductive cycle, trying to understand how it spread to new locations. What she found was remarkable. The fungus produced spores, as all fungi do, but these spores carried some of the archived genetic information with them, not the full sequences but fragments. Enough that if the spores encountered living members of the preserved species, they could potentially share that genetic material back. "It's not bringing extinct species back to life," Dr. Lopez explained carefully during a presentation, "but it might be able to help endangered populations by reintroducing genetic diversity that had been lost. Think of it as a genetic supplement for species under stress." The conservation implications were staggering. If the fungus could be cultivated and used deliberately, it might be possible to help struggling species recover genetic diversity they'd lost through bottleneck events or inbreeding. It wouldn't work on species that were completely gone, but for endangered species still hanging on, it could make a real difference. Environmental organizations took notice. Funding poured in from groups dedicated to preserving biodiversity. The University of Oregon became a center for this new field of research, what they were calling archival ecology, the study of how nature itself preserved genetic information from vanishing species. Margaret watched all this unfold with a mixture of pride and bewilderment. All she'd done was clean her shed. She'd found something unusual and called someone who could help figure out what it was. The fact that this simple act had sparked a revolution in conservation biology felt surreal. But late at night, when the phone stopped ringing and the emails stopped flooding in, Margaret would sit on her porch and think about what it all really meant. For decades, she'd watched species disappear. Every year brought news of another plant or animal gone forever. It had felt hopeless sometimes, like humans were destroying things faster than anyone could protect them. Now, though, there was a glimmer of something else. Not hope exactly. The fungus couldn't undo the damage humans had caused. But knowledge, understanding that nature had its own ways of remembering, its own methods of holding on to things worth preserving. The Luminomyces didn't think or plan or mourn. It simply did what millions of years of evolution had shaped it to do. It survived by collecting and protecting diversity, by building a library of life that might someday be useful again. Margaret looked out at her forest, the tall pines casting shadows in the moonlight, and wondered what other secrets were hidden out there. How many other mechanisms of preservation and survival existed that humans hadn't discovered yet? How much did they still not understand about the complex web of life? Her shed stood dark and quiet, its strange occupant long since removed to the lab. But Margaret knew she'd never look at that ordinary wooden structure the same way again. It had been a sanctuary, a refuge for the last members of a dying species, and in being that, it had become the birthplace of a new understanding about life, death, and the surprising ways that nature remembered its own. 3 years had passed since Margaret found the cocoon in her shed, and the world had changed in ways she never could have imagined. Her quiet farmhouse at the edge of the Oregon forest had become a landmark of sorts, not overrun with tourists, thankfully, but respected by the scientific community as the site where archival ecology was born. The county had designated her property as a protected research area, which meant she couldn't sell it or develop it even if she wanted to. Margaret didn't mind. She'd never planned to leave anyway. The designation came with some benefits too. The university paid for property maintenance and security, keeping away the occasional overeager journalist or documentary crew that still showed up hoping for an interview. The shed itself had been transformed. Instead of storing old garden tools and bags of fertilizer, it now housed climate monitoring equipment and motion-activated cameras. Researchers had discovered 3 more small fungal colonies on her property, all carefully documented and studied, but left undisturbed in their natural locations. Margaret's land had become a living laboratory. She'd gotten used to seeing graduate students tramping through her woods at odd hours, checking their equipment or taking samples. Some of them had been working on her property so long that she'd learned their names, invited them in for coffee on cold mornings. They treated her with a kind of reverent affection, like she was a beloved grandmother who'd accidentally discovered the secret of life. James had been promoted to dean of the science department, largely on the strength of the research coming out of the Luminomyces project. He'd gained weight and lost more hair, but his enthusiasm hadn't dimmed. He still called Margaret every week to update her on new developments, and she could hear the excitement in his voice even when he was explaining complex genetic data she only partially understood. Dr. Lopez had become the leading expert on archival fungi worldwide. She traveled constantly, consulting with conservation groups and governments about how to use the discoveries to help endangered species. Margaret had watched her give a TED Talk online, explaining to millions of viewers how nature had built its own backup system for biodiversity. The young scientist spoke with such passion and clarity that Margaret felt proud, even though she'd had nothing to do with Dr. Lopez's success beyond stumbling onto that first cocoon. The search for more archival fungal colonies had expanded globally. Teams were working in rainforests, grasslands, mountains, and wetlands across every continent except Antarctica. They'd found 17 different species of fungi that showed similar preservation behavior, though Luminomyces symbioticus remained the most complex and effective. Each new discovery added pieces to the puzzle of how these organisms worked and how they might be used to help struggling ecosystems. Some of the applications were already showing results. A project in Madagascar had used spores from an archival fungus to supplement the genetic diversity of a critically endangered lemur population. Early results suggested the lemurs were healthier and more resilient to disease. Similar projects were underway with California condors, Florida panthers, and dozens of plant species teetering on the edge of extinction. Not everyone was happy about the work, of course. Some environmentalists argued that the focus on genetic preservation was distracting from the real problem: habitat loss and climate change. If you didn't fix the underlying causes of extinction, they said, what good did it do to save genetic information? Others worried about the ethical implications of using fungi to manipulate species genetics, even with good intentions. Margaret understood both sides. She'd spent her career studying how human activity damaged ecosystems. The fungus wasn't a solution to that damage, but it was a tool, one that might help buy time for some species while humans figured out how to stop destroying the planet, or at least slow down the destruction. Her own life had settled into a new normal. She still gardened, still enjoyed her quiet mornings on the porch with coffee. But now she also gave occasional lectures at the university, talking to students about the importance of curiosity and careful observation. She'd become a symbol of citizen science, proof that important discoveries could come from anywhere, not just funded research projects in high-tech laboratories. The book had been a surprise. A science writer had approached her about collaborating on a memoir of the discovery and its aftermath. Margaret had been reluctant at first. She didn't think her life was interesting enough for a book. But the writer convinced her that the story wasn't just about her. It was about how ordinary moments could lead to extraordinary discoveries, how paying attention to the natural world could reveal wonders. The book, titled The Cocoon in the Shed, How One Discovery Changed Everything We Knew About Extinction, had been published 6 months ago. It sold better than anyone expected, especially for a science book. Margaret did a small book tour, speaking at libraries and bookstores across the Pacific Northwest. She met people who'd been inspired by the story to look more carefully at their own backyards, to report unusual findings, to participate in citizen science projects. One woman told Margaret she'd discovered a rare orchid species growing in her suburban yard after reading the book. A retired teacher in Seattle found what turned out to be a new species of moss in his local park. None of these discoveries were as dramatic as Margaret's cocoon, but they mattered. They added to human knowledge. They showed that nature still had secrets worth finding. Margaret had used some of. The book proceeds to establish a small scholarship fund for students studying conservation biology or mycology at Oregon State. It wasn't much, but it felt right. The Cocoon had changed her life in unexpected ways, given her a kind of late career prominence she'd never sought. The least she could do was help young scientists who were genuinely passionate about understanding and protecting the natural world. The original Cocoon, the one from her shed, was now permanently housed in a special museum exhibit at the university. Margaret had visited it once, standing in the dim viewing room and looking at the glowing structure behind protective glass. They'd managed to keep the fungus alive in a controlled environment, and it still pulsed with that eerie blue-green light. The preserved moths were visible inside, suspended in their crystalline tomb, beautiful and sad. A plaque beside the exhibit told the story of the discovery. It mentioned Margaret by name, explained what they'd learned about Pseudonarta flava and Luminomyces symbioticus, but what struck Margaret most was the last line of the description. This discovery reminds us that nature is more resilient and creative than we often imagine, and that extinction, while tragic, may not always be the final word. That thought stayed with her as she drove home from the museum that day. Extinction wasn't the final word, at least not always. The fungus had found a way to remember, to preserve, to hold on to something precious even as it slipped away. It wasn't